Shri Gurubhyo Namaga children. I am happy to welcome you all for our online teaching academy, Ekalaiva. For the class 11, subject Biobotany, chapter 1, Diversity of Living World. Today, we are going to discuss about multiplication or life cycle of phage. In the previous class, that is the last class, we discussed about bacteriophage. What is bacteriophage, children? A virus which infect bacteria are called bacteriophage. The good example is, yes, very good, T4 bacteriophage, right? What is the shape of the bacteriophage? What is the shape of that virus? Tadpole shape. So all the things we discussed yesterday. Today we are going to see the multiplication of phages. Phages multiply through two different types of life cycle. The first one is Lytic Cycle and the second one is Lysogenic Cycle. The Lytic Cycle is also called as Virulent Cycle and Lysogenic Cycle is also called as Avirulent. How you have to pronounce? Yes, Avirulent. Virulent means poison. Okay. Avirulent means non-poison. So, phages multiply through two different types of life cycle. One is Lytic cycle, another one is Lysogenic cycle. Clear, children? Yes. The first one is Lytic cycle. During Lytic cycle, the phages disintegrate. What is the meaning of disintegrate? Think. Disintegrate means destroy, okay, damage. So the phage disintegrate the host bacterial cells. Host bacterial cell here is E. coli. This is a host bacterial cell. Bacteriophage disintegrates this host cell and the progeny, the virus that is called the viron are released out from here. So this is a virus called bacteriophage. This bacteriophage virus disintegrates the host bacterial cell and finally progeny, the virons are released out. Steps involved in Lytic cycle are, there are 5 different steps. The first one is adsorption. What is adsorption? Second one, penetration. Third one, synthesis. Fourth one, assembly and maturation. The last one, release. Can you tell once again? Yes, adsorption. What is the meaning of adsorption? Adsorption is nothing but attachment. As I said, no, the phage virus, bacteriophage attached on the walls of the bacterial cell. The first step is adsorption, not absorption, adsorption. Second step, penetration. The genetic material of this bacteriophage slowly enters. Penetrate means enters. And third one, synthesis. Synthesis is nothing but formation. And then fourth one, assembly. They get ready to come out from the bacterial cell. The last one is release. So these are the 5 steps involved in Lytic cycle. Clear, children? The first step is, as I said, adsorption. See here, see the picture here, children. What is this? This is a bacteriophage. This is the bacteriophage. The bacteriophage example is T4 phase, T4 bacteriophage. The T4 bacteriophage intact with the cell wall of the host. This is the host, cell wall of the host. Example is E. coli. E. coli is a example for bacteria. Clear? So this is the host organism. The virus, the bacteriophage come and contact with the host organism. They attached on the cell wall of the bacterial cell. Can you get my point, children? Yes. What happened? The phage tail, you know very well, bacteriophage have different parts. What is the shape of the head part of the bacteriophage, children? Yes, hexagonal head. So this is the tail. This part is called collar, and this is the head portion, head, hexagonal head. Or get my point, children? Yes. And this is the tail fiber. The same is there here also, tail. So the phage tail makes contact with the host cell. That means host cell membrane. Clear? And the tail recognize the specific receptors. The bacteriophage do not go and attached in some other place. It always attached in the receptors which is present on the cell wall of bacteria. So the bacteriophage go and recognize the search where the receptor is present. Once it search means, then this particular bacteriophage go and attached on the receptors of the bacterial cell. See here, the phage tail makes contact with the tail fibers and recognize the specific receptors present on the bacterial surface. Bacterial surface is nothing but cell wall of the bacteria. Get my point, children? So this is called adsorption. As I said, adsorption is attachment. Initially this bacterial cell, this is called virus, bacteriophage. This virus attached on the surface of the bacterial cell. That process is called adsorption. This is the first step in Lytic cycle. The lipopolysaccharide of tail fibers. This is a clear picture of bacteriophage, hexagonal head. So this is the tail fiber. The lipopolysaccharide of tail fiber act as receptors in phage. So this receptors goes and bite attached on the receptors of the bacterial cell. Clear? Yes. The process involving in recognition to bacterium is called landing. So recognition of phage to bacteria, that is called landing. Landing is nothing but come and attach. Okay. See here, the process involving in the recognition of phage to bacteria is called landing. Once the contact is established, once the contact is taken place between the tail fiber and the bacterial cell, the tail fiber bend to anchor. Now the tail fiber becomes very short. It bends to anchor to the cell surface. This is said to be pinning. Pinning is nothing but bending of tail fibers and anchor the pins and the base plate to the cell surface. Cell surface is nothing but bacterial cell surface. Clear, children? Yes. The second step is penetration. Penetration is nothing but entry of nucleic acid. Penetration is nothing but entry. Yes. So penetration is a process involves mechanical and enzymatic digestion of cell wall of the host. At the recognition site, phage digests certain cell wall by viral enzyme called lysozyme. Very important. See here, this is the bacterial cell and the phage. The phage which is attached on the receptors of the bacterial cell. Once the attachment is established, then the next one, the entry of nucleic acid takes place. The phage digests the cell wall. Digestion means here the destruction of cell wall takes place and the enzyme, with the help of the enzyme lysozyme. Okay. So after pinning, the tail sheath contracts and appear shorter and thicker. Okay. So after contraction of base plate through which DNA is injected, see here, this is the first step, attachment of bacteriophage on the receptors of the host organism. Here host organism is nothing but E. coli. Once the attachment is over, attachment is established, the second step, the entry of nucleic acid takes place. After pinning, the tail fiber became thicker and shorter, so the destruction of the cell wall takes place. Now the nucleic acid slowly enters into the bacterial cell. This is the bacterial genome and this is the viral genome. Viral genome is injected into the cell. What type of cell? Bacterial cell. This is a bacterial cell, this is a virus. Clear children? See here. After contraction of the base plate enlarged through which the DNA is injected, it enters. Okay. The step involving injection of DNA particle along into the bacterial cell is called transfection. So transfection is nothing but injection of DNA particle along into the bacterial cell. So transfection is nothing but entry of nucleic acid, entry of DNA particle into the host cell, into the bacterial cell, that is called transfection. Third step is synthesis. This step involves the degradation of bacterial chromosome and DNA replication. So these steps involve in degradation of bacterial chromosome, protein synthesis and DNA replication. You know very well, the E. coli is a prokaryotic cell. The E. coli is a prokaryotic organism, the cell is a prokaryotic cell. So they have a DNA and then they have a plasmid, extra chromosomal circular DNA plasmid is there, and then they have a replication property. So once the virus enters into the bacterial cell, it degrades, it arrests the protein synthesis, DNA replication, etc. The phage nucleic acid, what type of nucleic acid present in bacteriophage? Yes, exactly, DNA. So the phage DNA takes over the host biosynthetic machinery. Once the DNA enters into the host organism, that means into the bacterial cell, the phage DNA, what happened? The phage DNA takes over the host bacterial machinery. Okay. So host DNA gets inactivated. So bacterial DNA, host DNA is nothing but bacterial DNA gets inactivated and breaks down. So the viral DNA, what type of DNA? Double stranded DNA. The viral DNA takes over and attack the bacterial DNA. So the host DNA get inactivated and breaks down. Now the phage DNA suppresses the synthesis of bacterial protein and simultaneously the replication of phage DNA also takes place. So the phage DNA suppresses the bacterial DNA, automatically then the phage DNA get ready for replication. Replication is nothing but exact carbon copy of DNA. Okay. The next one is assembly, fourth step, assembly and maturation. The DNA of the phage and protein coat are synthesized separately and are assembled for phage particles. The process of assembling the phage particle means phage DNA, that means normal virus. Phage particle is known as maturation. After 20 minutes of infection, about 300 new phages are assembled. See the picture here, this is bacterial cell E. coli. This is bacterial genome. Bacterial genome is nothing but bacterial DNA. Clear, children? What is this, children? This is a bacteriophage. It is a virus. Virus containing the nucleic acid is DNA. Once the virus attached on the surface of the bacterial cell, that process is called attachment. After that, the viral DNA slowly enters into the bacterial cell. That process is called penetration. So this is called phage DNA. This phage DNA destroy the bacterial DNA and produce so many virions. So synthesis takes place. After that, they get ready, they assemble together, they form a matured virus, bacteriophage, and then they are ready to come out, the release of new phage particle. Fifth step, the phage particle gets accumulated, they get ready inside the host cell. Inside the host cell is nothing but bacterial cell, and are released by lysis of host cell wall. They are ready to come out. Once it is ready, it lies the cell wall of the bacterial cell. Lysis is nothing but breakdown of cell wall. So this bacteriophage breaks the cell wall of bacteria. This is the bacteriophage. This bacteriophage lies the cell wall of bacterial cell and come out as a many number of bacteriophages. Can you get my point, children? So release means nothing but the phage particle. This is the phage particle gets accumulated. They are all get accumulated and accumulated inside the bacterial cell, and they are ready to come out and are released by the lysis. Lysis is nothing but breakdown. Lysis is nothing but breakdown of cell wall. And finally, the phage particle come out, the release of new phage particle. Clear, children? The next cycle, as I said, Lytic cycle and Lysogenic cycle. Now we are going to discuss about Lysogenic cycle. Lysogenic cycle take nothing but the phage DNA. You know very well, bacteriophage having the nucleic acid DNA, that is called the phage DNA, gets integrated into the host DNA. Get integrated means it goes into the host cell and integrated with the host DNA and multiplied along with the nucleic acid of the host. See here, I am going to draw a simple diagram here. What is this, children? Think that it is a E. coli. Clear, children? E. coli. What is this, children? Bacterial genome. Correct? Yes. Now, what is this, children? Phage that have another type of genome, that is phage DNA, viral DNA. Once the viral DNA enters into the bacterial cell, it migrate inside, it penetrate inside and it integrated with the bacterial genome. So the viral genome integrated with the bacterial genome. This is viral genome. This viral genome integrated with the bacterial genome. That is the only difference between Lytic cycle and Lysogenic cycle. Clear, children? See, no independent viral particle is formed. As soon as the phage inject its linear DNA, so the phage have the structure linear. Here the DNA is linear. Once the linear DNA enters into the bacterial cell, it integrated to become a circular DNA. Clear children? See, so this is linear. Once it enters, it forms the circular DNA. As soon as the phage inject its linear DNA into the host cell, it becomes circular. Very, very important. Okay, and integrates into the bacterial chromosome by recombination. So the integrated phage DNA is now called as prophage. The activity of prophage is nothing but integrated DNA. Okay, so the activity of prophage gene is repressed by two repressor proteins which are synthesized by phage genes. This checks the synthesis of new phage within the host cell. However, each time the bacterial cell divides, whenever the bacterial cell divides, automatically the prophage also divide into two. See here, the bacterial cell divides, the prophage multiplies along with the bacterial chromosome. Clear children? So, on exposure to UV radiation, ultraviolet radiation and chemicals, the excision of phage DNA may occur and results in Lytic cycle. Clear children? See the diagram here. You know very well, this is the bacterial cell, E. coli. This is a phage, bacteriophage. This is a viral genome. This is a bacterial genome. This violet color is bacterial genome. This is the blue color is viral genome. Once it enters, this is again the viral genome. See here, this is a linear structure. This is circular structure. Clear children? So, and then the integration takes place. This is called prophase, recombination of the viral DNA and the bacterial DNA. Once it multiplies, the bacterial cell multiply, the prophage also divides, multiply. This is called the lysogenic cycle. Next, two more question. Virions. Virion is an intact infective virus particle which is non-replicating outside the host cell. Virioid is nothing but circular molecule of single stranded RNA without a capsid and was discovered by T.O. Dainer in the year 1971. The RNA of virioid has low molecular weight. Virioids causes citrus exocortics and potato spindle tuber disease in plants. Thank you children, we will see you in the next class.